Well, good afternoon, and thank you all for joining us today for Cancer Center Grand Rounds. We're really very fortunate to have a special speaker here today because, as many of you know, today we are recognizing the Blanche Tolman Lecture Series. For those of you who aren't aware, the Blanche Tolman Series was endowed by Dr. Marvin Sears, Professor Emeritus at Ophthalmology in memory of Dr. Sears' mother, Blanche Tolman. And that lectureship has brought to our center extraordinarily talented individuals focused on research, particularly in the realm of hematology and hematologic malignancies. And we're very fortunate to continue that tradition today with our invited speaker, Dr. Michael Caligiuri. Dr. Caligiuri is the president and physician-in-chief of the City of Hope National Cancer Center, and who Mike's career as a physician scientist, a leader, executive, mentor is really unparalleled. Mike was for 19 years at the James Cancer Hospital in Ohio State, and ultimately the CEO of the James, where he took that institution to an altogether new level in terms of clinical care, research, philanthropy, recruitment, and really bringing it to a great impact in cancer care regionally, nationally, and internationally. As well, most recently then took on this new role at City of Hope. Mike's research has had an enormous impact on our understanding of leukemia and lymphoma. Recognizing his research, he was elected to the National Academy of Medicine in 2018, and recognizing as well his leadership in research, he is a past president of the American Association for Cancer Research. Among Mike's many accomplishments, I was particularly impressed that he has trained over 120 undergraduate, graduate, and postgraduate students who've now gone on to receive 230 grants and awards. I think an extraordinary accomplishment for a mentor. So I'm very pleased to welcome the 2019 Blanche Tolman Lecturer, Dr. Michael Caligiuri.
Very nice, sir. Thank you. Charlie, thank you very much. Very kind introduction. I had the pleasure of having Charlie as a senior resident when I was at the Brigham, and he continues to do amazing things. So it's a pleasure to be here. It's so good to see so many friends, and so nice of all of you to come on such a snowy day. Hey, Roy. We were supposed to meet this morning, but you got busy.
We'll talk later.
Good. I realized some of you might have kids to pick up. School closing early, and then I heard that school was already closed yesterday for today. So that's impressive preventative medicine. I'm from Buffalo, so this is like we used to row in this kind of weather in the canal, you know. But it's a great pleasure, and it's an honor to be here. And especially because I'm going to talk about natural killer cells, I see that there's a great deal of interest, and that's really neat for me because not a lot known when I started in this field. And what I want to do is really take you through kind of my story of different discoveries and translation that we've made with natural killer cells through the years, and finish with some clinical application. I'm not going to talk about its application in acute myeloid leukemia, which is probably perhaps the most exciting, but probably the best known, but really where we're going with natural killer cells as we learn more and more about their biology. My lab is really focused since the beginning is on understanding receptors, because if you understand receptors that sit on the surface of a cell, you can understand what that cell sees, and if you understand what that ligand binding to the receptor is doing, function, really start to think about how do we manipulate these to help in cancer and infectious disease. So first, just some disclosures here of mine. And I really won't be addressing any of this today. And so we'll talk about role of cytokines in natural killer cell development, secondary lymphoid tissue, which is where we've discovered NK cells develop. We'll talk about subsets and clinical applications of that receptor biology. So back a long time ago, this is what was known about natural killer cells. You know, this is cover of Time magazine. You could take these CD34 stem cells and pop them into interleukin-2, and after about 14 days to 3 weeks or 4 weeks, you get natural killer cells. And really described by this CD56 bright cell, which is an immature NK cell. As it becomes dim on the surface of the cell, it's a mature NK cell. And we'll hear about CD56 bright and dim. But really the question was, is this the real growth factor for IL-2 in vivo in our bodies? Because we knew that IL-2, while a lymphocytotropic hormone, was very selectively released by T cells on activation, and yet you have billions and billions of NK cells in your body, and turnover is fairly rapid. So Mother Nature helped us with that with some genetic disruption experiments, because the mouse, when knocked out with IL-2, didn't have NK cells, and the mouse knocked out of the IL-2 alpha chain, which is one of 3 chains of the IL-2 receptor, had NK cells. Both the IL-2 knockout and the IL-2 alpha chain knockout had NK cells. However, if you knocked out the beta chain of the IL-2 receptor or the common gamma chain of the IL-2 receptor, there were no NK cells. So that told us that something that isn't IL-2 and doesn't require the alpha chain, but does require the beta chain and the gamma chain, is important for NK cell survival. And so that receptor schematically looks something like this, where you see the beta gamma, and those are the signal transducing chains. That's where all the action is. But for that beta gamma to see IL-2 in your body, it needs that third tiny alpha chain. That alpha chain, while not signal transducing, confers very high affinity for physiologic doses of IL-2, and with that transduces a signal. But by itself, the beta gamma chain can transduce a powerful signal, but just requires enormously high doses of IL-2, and that's why IL-2, when given exogenously in high doses, can make NK cells. And so what was subsequently discovered was another molecule that did precisely what I told you. It had its own alpha chain, IL-15 receptor alpha chain, and unlike the alpha chain of IL-2, the alpha chain of IL-15 by itself can bind IL-15 with very high affinity. And it sits on other cells, antigen-presenting cells, and it presents the molecule IL-15 to the beta gamma chain, and that sends a signal. And then we can see this here, when we take IL-15, pop it into culture with those CD34 positive cells, and now we're looking at a histogram here, and you can see the CD56 bright cell, which is the immature NK cell. What we noticed was that even though there were pure cells from this uncommitted hematopoietic progenitor cell, there were very few in number with IL-15 alone. And so we asked ourselves, what other receptors might there be in the CD34 cell that augments the growth of these? And so to do that, we actually looked at this cell. So what receptors are on this cell that might tell us a little bit more about that? By the way, IL-15 knockout mice and IL-15 receptor alpha chain knockout mice lack NK cells, confirming what we found in humans. All the work I'm going to talk about is in humans, and then I refer back to the mouse for studies we go to the mouse for, but we work primarily in human natural killer cell development. So to look at what else could be involved in the growth of NK cells, we went back and we found that this receptor tyrosine kinase, c-KIT, was also on the surface of just the bright NK cells, not on the surface of the more abundant dim NK cells that are floating around in your blood, but when collected on the bright NK cells, you can see fading as the cells become dimmer. And so we looked to see if KIT ligand, stem cell factor also known, or mast cell growth factor, and its sister FLT3 ligand could be important in NK cell development. And so here you see KIT ligand or FLT ligand. Here's total cell number from those CD34 cells initially, and here's the NK cell numbers. So KIT ligand and FLT ligand alone with CD34 cells gives you growth. Here we're looking at FL alone, but as you can see, there's no natural killer cells, though no natural killer cells here. IL-15, I showed you, very high numbers of NK cells, very low growth. Then so together, very low growth of NK cells. But combined, KL or FL, when combined with interleukin-15, and both of these stromal cell factors, shows that not only do you get the purity of NK cells, but you get synergy and growth of total cell numbers, so you get high numbers of cells here. So the way that FL and KL work on the NK cell without affecting it by itself is twofold. One is through the proliferation pathway, which I'm not showing you, but the other is it upregulates the beta and gamma chain on more CD34 positive cells, allowing a greater number of those cells to be responsive to interleukin 15. So the picture then began to unfold as such, where an earlier cell, a progenitor, has on its surface KIT and or FLT3 and sees FLT3 or KIT ligand and pops up IL-15 receptor beta and gamma chain, and then in the presence of IL-15 would produce the natural killer cells. And exogenous IL-2 in high doses could do this same thing. So this was interesting. So we wanted to go after this cell to see if we could identify the true natural killer cell precursor in humans, because if we could start to figure out the developmental pathway beyond this. We could figure out a couple of things. Where do human NK cells develop? And then what are the molecules involved in their development? So in looking for this cell, we said, well, let's just look for this IL-15 receptor beta and gamma chain on the surface of the CD34 cell. We couldn't see that by flow cytometry at all, but we knew it had to be in the 34 population because of this pathway. So what we did is we took the 34 positive cells and using a variety of markers I'm not showing you, found 3 different populations from these 2 markers, CD45RA and 34, 1, 2, and 3. And we popped each of those into culture with IL-15 or high-dose IL-2. And you see only the 3rd population over here with number 3 labeled here, CD34 low, RA positive, was the one that became natural killer cells. And indeed, we further took that population and said, what other receptors are on the surface of this population to tell us where this cell might live? What we found uniquely was that this population of CD34 positive cells expressed high levels of L-selectin and beta-7 integrin. And these 2 molecules were known at this time to be important for homing to secondary lymphoid tissue, such as tonsil and spleen and lymph nodes. So there'd never been CD34 cells found in any of these tissues up until this time, but we decided to take a look. And so we first, to make sure our system worked, we had a CD34 column. We put blood through CD34 column, and you see all 3 populations, when enhanced through this selective column found in your circulating blood. When we did the same thing with a collection of human lymph nodes or tonsil, you can see that we only see this population. So the receptor biology brought us to the right population. And in fact, when you take that population out of the fresh lymph nodes or tonsil and you put it in IL-15, you see you get your CD56 bright natural killer cells right here. So that was really interesting. And then we said, well, why are they going to lymph nodes? And we looked at lymph nodes and we found that there were CD56 bright immature NK cells in the lymph nodes as well. And they lived around the parafollicular T cell rich region of the lymph nodes. And so we asked ourselves, well, if you have the early NK cell here and you have the earliest precursor here in secondary lymphoid tissue, maybe this is where human NK cells actually develop. In mouse, they develop in the bone marrow, and that didn't seem to be the case in humans. So a lot of work, and by the way, I'm showing different pictures of people that are responsible for the work in the lab. We've narrowed this down to 3 different markers: acquisition of CD34, no c-KIT, no CD94, CD34 with c-KIT, no CD94, loss of CD34, c-KIT and CD94 negative, and then just CD94 positive. When you took these populations, you said, let's look at CD56, and this is from lymph nodes, you see that we see absent, some, a lot, and then CD56 bright cells. So we named these stages 1, 2, 3, and 4 of NK cell development. We could find these all fresh in lymph nodes and tonsil. And it's been a really interesting journey characterizing these various stages of NK cell development for a number of reasons. One reason is when we characterized this population here, we found out that actually before it transforms to an NK cell, it's actually what's called an innate lymphoid cell type 3, ILC3s. ILC3s are very important in gastrointestinal physiology and pathophysiology, secreting abundant interleukin 22, which drives the epithelial cells of the gut to secrete defensins for protection against bacterial invasion. When this cell actually loses a transcription factor called AHR, it drives all the way to a natural killer cell. So this ILC3 and NK cells actually live in balance depending on which tissue they're in. But the picture began to sum up something like this, where T cells developing in the thymus, B cells in the bone marrow, and NK cells having a progenitor that left the bone marrow, went to secondary lymphoid tissue, and then really proceeded in this pathway here. And over the years, just to summarize work from our lab predominantly and some work from other labs, we've gone on to further dissect how not only NK cells develop, but how human innate lymphoid cells, of which there are at least 3 types, develop within secondary lymphoid tissue with a variety of different types of discovery. And I'll just focus on one of those. Here you see we've learned that we go from a progenitor here to about 3 or 4 different stages. Here we get a progenitor for both ILC3 and NK. And when you get to this stage 4A, it's quite a unique population. I'll show you how this works. So I mentioned you acquire CD117, that's shown here. Then you acquire NKP80, and then you acquire CD16. And once you get to CD16, you've got a mature NK cell that's floating around in your blood. Remember, CD16 is the receptor for immunoglobulin. It's what's important for Rituxan and Herceptin for mediate antibody-dependent killing. And this all occurs through these in situ, these pathology flow cytometric pictures of normal lymph nodes in tonsil as well as all your lymph nodes. And of course, you have kilograms and kilograms of your lymph nodes, so lots and lots of these cells are developing. What's interesting is that this stage 4A, as you've seen here, we actually have found something very interesting to show you where this work goes into the clinic and into the lab. And so if you look, it turns out here's you see 4A is only in tonsil or lymph nodes. You don't see it in blood or bone marrow or other places as well. What we found is that there's a malignant counterpart to stage 4A, and that's this EBV TNK cell lymphoma, this god-awful tumor that you see here. And it turns out that if you look at 4 different patients with this, and you look at their tumors, whether it's blood, bone marrow, blood here, or central nervous system with CNS involvement, all of them have their malignant counterpart in this stage 4A, originally found as part of the NK cell development paradigm. So we're actually doing lots of work on the genomic and epigenomic differences between these normal and malignant populations, have some very interesting findings, and moving this out for publication soon. So now you're experts on NK cell development. There's no quiz, but I'm sure you all get it now. We'll talk a little bit about survival and what goes wrong with NK survival. So once we understood that IL-15 was critical for the development, we asked, how do NK cells sustain? Just like your hemoglobin, they remain very, very constant in your body despite tremendous dynamics. And we asked if IL-15 could be the factor responsible for that. So just taking NK cells and placing them in IL-15 alone, serum free media, you can see that a tiny amount of IL-15, 0.01 nanograms per mil, will sustain their survival for over a week. So that gave us a hint, but it's only, of course, an in vitro experiment. To do this properly, you'd want to take normal NK cells, pop them into a mouse that has no IL-15, and see if those NK cells, which can't develop in that mouse, still survive in that mouse. And so we did that experiment. Here's your normal NK cells from a wild-type mouse, coat them with a dye, put them into a knockout mouse and say, do they survive? And the picture tells the story, because here's normal mice going into normal mice. So you took them out, you labeled them with dye, and 36 hours later, you see their 0.12 is what's in there. And a week, 5 days later, it's 0.11, essentially the same. You do this into an IL-15 knockout mouse, the wild-type cells into an IL-15, and within 36 hours, you're drastically down, and within 5 days, they're completely gone. So this told us that IL-15 is likely very, very important for not only NK cell development, but NK cell homeostasis, survival in vivo as well. So where's the IL-15 coming from? Obviously not the marrow. These aren't developing the marrow, something in the lymph node. And we figured this was an antigen-presenting cell, most likely a dendritic cell. So we ended up doing an experiment, again, going to the mouse to prove that when you knock out dendritic cells, which I show here, this is the dendritic cell in the mouse, and now they're knocked out through this mechanism, actually NK cells are drastically reduced, if not almost absent. So the picture is FLT3 ligand is important not only for that CD34 cell, but of course it sustains the survival and the growth of dendritic cells. It induces IL-15 or sustains IL-15 on a membrane surface through that IL-15 receptor alpha, that high affinity chain. And then that, with precursors, leads to NK cell development. And then as NK cells develop and trickle through your secondary lymphoid tissue constantly, they get a hit from IL-15 and continue with their survival. What's interesting about IL-15 is that the mRNA is in virtually every tissue in your body. It's abundant in brain, heart, liver, kidney, but very, very little protein anywhere. And so its modification, it's impossible to detect most methods in serum, for example. So one of the things it must be is that it's being translated post-translational regulation, post-transcriptional regulation. And so we looked at that, and you can see here that what we did is we did a little jockeying with this gene and put basically some constructs into the gene to make it so that the transcript, the abundant transcript, was transcribed, and not only transcribed, but abundantly secreted. And developed this transgenic mouse simply to ask the question, when you alter this cytokine, what happens to NK cells? As you can imagine, NK cells are abundantly increased as shown here. Here you're looking at a mouse, and you can see a wild-type mouse has this much NK. Same mouse, it's transgenic for the IL-15, where it's getting abundantly transcribed, translated, and secreted, has huge numbers of NK cells. So very predictable. Driving development, sustaining survival, you're going to get lots of NK cells. What was perhaps more interesting, however, is this is after about 3 to 6 months, these mice developed TNK cell, LGL leukemia. I think it was the first time a single cytokine manipulation was shown to be oncogenic in a mouse. And these mice developed massive leukocytosis, sometimes 500,000-600,000, and would die from overwhelming accumulation of these blasts in the body. And as I say, it was NK cells, as shown here, but also NKT, which is another innate immune effector cell. And when we looked at the NKT cells, we could determine if it was clonal by looking at the T cell gene rearrangement. And we could show here that it was in fact clonal. Here's an example of the lymphoblasts. You can see prominent nuclei, prominent nucleoli, and very blast-like picture, different from the large granular lymphocyte in normal times. Further, very easy to adoptively transfer and lethal in the syngeneic mice. So that was interesting, and especially because we also study AML, which I'm not talking about today, but the leukemiologists here know that about 30% of AML and some ALL have constitutive activation of FLT3, the receptor tyrosine that I talked about earlier. And when the ITD is present, that receptor is constitutively active, as if there's abundant ligand always activating the receptor. And that's at least a precursor to malignant transformation into AML and sometimes ALL. But fully 30% of AML has this particular mutation, usually requiring another cooperative mutation. It was interesting to us because we didn't have a constitutively active receptor like the ITD, but we had constitutively active ligand in this transgenic mouse. So it was like the IL-15 beta gamma chain was always on. And sure enough, we saw this, with just this manipulation alone, we saw this transformation. So this became interesting and relatively easy for us to study. And I'll make a long story short about that. Kind of working backwards, we noticed that the blasts in both human LGL and our mice with this LGL leukemia had aberrant and abundant centrosomes. And we started to look at the pathway that's important for centrosome development and maintenance, and it involved MYC. And so to summarize, what we determined was that IL-15 was driving NF-kappa B to increase MYC, and then together we're driving up aurora kinases A and B, and that led to chromosome instability through this centrosome aberrancy, and part of the story for leukemia genesis. What we also found, which was interesting, was that NF-kappa B and MYC were teaming up with HDAC1, and this trimer repressor complex popped down and drove miR29B to the depth, which miR29B regulates DNMT3B, and the leukemia docs know this is often mutated in AML. And by low miR29B and high DNMT3B, we got tremendous amounts of methylation at the 5' regulatory region of genes, which led to further chromosome instability, gene silencing, and leukemia. So put this pathway together for the genesis of LGL leukemia through overexpression of IL-15. And to focus on this therapeutically, we had determined that bortezomib actually broke up this repressor complex and could lead to driving miR29B up and DNMT3B down. And so we focused on that, and here you see the simplistic schema where you have bortezomib hitting on the repressor complex, disappearing miR29B going way up, driving DNMT3B way down. And so we treat the LGL leukemia with bortezomib. You see the DNMT3B disappears up here in this gel. So it seemed to be working. We took this then to the clinic and had huge success. Kidding. But didn't give up. Actually turned out the PK was off. We ended up developing a nano bortezomib and ended up showing that in our animal model, we could get 100% cure of this LGL leukemia with this nano bortezomib. And this gentleman, still at OSU, is further developing this compound for the clinic. The other thing I just thought I'd show here is very interesting. Angeli came in my lab and said, so that happens in 30% of the transgenic mice. What happens to the other 70%? Of course, I'd never been to the mouse room, so I said, I don't know. So she went to the mouse room, and a long story short, she found that these mice were getting this progressive alopecia and ultimately this very serious cutaneous problem, denuding of the fur, et cetera. And again, telling a long story in a single slide, this turned out to be a great model for CTCL, cutaneous T-cell lymphoma, something studied so incredibly well here at Yale. And as you can see here, the pathology is classic for CTCL, and I could replace these with normal and CTCL patient, and quite indistinguishable. The infiltrate is a CD3 null or a CD3, CD4 T cell. And again, a lot of work being done comparing this in this mouse model, and we've got great strategies now on their way to the clinic to both slow down or prevent the disease. Very exciting epigenetic modifiers. So that's a bit about NK cell survival, deregulation. By the way, IL-15 is abundantly overexpressed in CTCL, and you may now know there's a peptide being tested that blocks IL-15 binding, and they've had some, maybe it's open, the trial's open here, but there have been some dramatic responses in multiply relapsed CTCL, suggesting that what we're finding in this transgenic mouse is quite important in the human pathogenesis of the disease. So moving on to a little more defensive, what are these NK cells normally doing in the body? Here you see, this is a typical flow of your blood. Your T cells and B cells are down here. Your NK cells, a few of the immature ones up here, the bright cells. And as they become dim, they acquire Cd16, which is that molecule that hooks up with antibody to do the ADCC killing. And so one of the things we were wondering is, does the bright cells floating in your blood versus and they're largely in the lymph nodes, and the dim cells, which you don't find in the lymph nodes but are in your blood abundantly, do they have different immunoregulatory properties? And so one of the things we knew is that NK cells can make gamma interferon in a heartbeat, like within a minute, if they're tweaked the right way with monokines, IL-12, IL-18, IL-1, IL-15, they can make lots of gamma interferon. And we knew that monocytes need gamma interferon to contain obligate intracellular pathogens like TB and pneumocystis and listeria, et cetera. And so we said, well, if they don't make gamma, they must get it early on from NK cells. And could there be a difference between the bright cell and the dim cells to what cell's providing that monocyte macrophage with that requisite gamma interferon to contain these infections? And we know these infections are contained because people that lack either the gamma receptor or gamma interferon die of overwhelming obligate intracellular pathogens such as tuberculosis. So we asked, are they the same or different? We set up an assay where we put monocytes in a well, in either the bright cells or the dim cells, and then we added the pathogen, and to see monokines delivered, is the bright cell or the dim cell the cell that's delivering the gamma interferon? And it turns out, as you can see from this slide, even when you just give the monokines or you do that assay I showed you, it's really the bright cells that are making all of the gamma interferon. The dim cells that are more abundant in the blood aren't. And that made more sense to us because, of course, the dim cells live in the blood and the brights largely live in that parafollicular T cell rich region of the lymph node. So one model is within your lymph node, pathogens enter, NK cells are tweaked with monokines, they make lots of gamma interferon, there are more monokines produced, and it's a very nice cycle. We also learned, and I'm not developing the story here at all today, is that in addition to that, those NK cells that sit in the parafollicular T cell rich region of lymph node actually get a signal from T cells as well. And they get it from that tiny amount of interleukin 2 that the T cells secretes when it's activated. Because just the bright cell, not the dim cell, has the high affinity IL-2 receptor on its surface in addition to the beta gamma cell. So it can compete for IL-2 with the T cell. So if it gets the antigen presenting cell, for example, gives 12 and T gives 2, we've shown this in our culture constructs, you actually get abundant gamma interferon that can drive this to a Th1 important adaptive immune response to certain pathogens. So the NK cell is involved in both of those. The bright cell, I should say, is involved in both of those in its niche in the secondary lymphoid tissue where it sits. So that's here, bright cell. Doesn't have many granules. Again, receptors tell the story, produces lots of different cytokines to stimulate the innate and adaptive immune system, certain receptors that suggest homing, et cetera, and very little Cd16, as I say, very little granules, so not a killer. In contrast, the dim cell, which is floating around in your blood, abundant granules, lots of different receptors. Here's the KIR immunoglobulin receptors, which are an inhibitory signal largely, tells NK cells don't kill, and I'll refer to that a little later. Very few cytokines are produced, as I showed you, and abundant Cd16 to bind all that antibody. And so there's been a beautiful story developed when the KIR is mismatched in allogeneic transplant. Many of you hematologists know that story. I'm not going to talk about it today. It's fascinating. There's increased survival in patients that undergo full T-cell depleted mismatched transplant that have KIR mismatch as well. I'm going to talk about the Cd16 receptor largely today. I'm going to refer back to these inhibitory receptors for a moment and talk about some discoveries we've made and how we're taking them to the clinic about Cd16, which, as I say, we know is important. So we'll try to get inside of the mind of the killer. This is an NK cell. This is the tumor cell. It's puncturing holes from its perforin release, and then the cytoskeleton of the tumor. And the NK cell recovers after that, can regenerate and kill again. So very interesting tumor lysis. So I'm going to talk about glioblastoma. Bad tumor, as many of you know. Very fatal. We've all known friends and family that have had this devastating disease, horrible survival with this. We've been working on this in our lab now for a while because I teamed up with a neurosurgeon, Kioka, was at Ohio State, and he kept noticing that there were gamma interferon producing cells when he would try to treat GBM with oncolytic virus, oncolytic herpes virus. And I'm going to tell you how that story's evolved for us. So just to digress, we do have a CAR T program, very active, and we'll soon have CAR NK for GBM at City of Hope. And this has been very, very interesting study that continues. So here's what happens with oncolytic virus. There's a couple videos in here. And just to show you, so what we do is you create a virus that only becomes lytic in the context of a tumor-specific promoter, which is found in the GBM and not in your normal brain tissue. So herpes virus, as you know, is a neurotropic virus, right? Kids die of herpes encephalitis. Adults die of herpes encephalitis. It's attracted to the nervous system. So it's a perfect virus to consider for therapeutics because it goes to neural tissue. And with the right promoter, it'll only become lytic in a GBM versus normal tissue. So what happens is the virus comes along, it'll infect one cell, and then what's critical is that that virus is able to spread along. It spreads along, and as it spreads along, it then becomes lytic. So it needs time to do this. It needs time to infect, and it needs time to spread before it will become lytic. The problem with this, and why my friend Nino Kioka called me, is that it turns out NK cells protect you against dying of herpes. Anybody gets a cold sore, it's a limited infection. It's not Ebola, right? Whatever you have an infection, but you don't have any B cell or T cell immunity to it, your first exposure, but it's very limited. And it turns out that NK cells are critically important to limit that infection, as evidenced again by Mother Nature and the problems we have when NK cells aren't around. And this has been shown in several different examples, but how and why they work was really unknown. So what we found, Nino kept calling me, the gamma-producing cells, is that the NK cells were flocking into the brain tumor before the herpes virus got a chance to work on the brain tumor. Massive numbers of NK cells going in there and killing the virus. And I'll just illustrate that. So here's your tumor in the context of normal neural tissue. You get your herpes virus comes in. It's going to, again, go to infect your GBM and start its process of spreading. Before it even gets a chance to spread, your NK cell is there to kill it, such that it's ineffective. And we showed in rat models, if you deplete NK cells or mouse models, you can get great response to the brain tumor. So we knew we had something. But understanding what to do really had to understand what's going on here to really start to, and no clue, despite that other paper, I think, being published in the early 90s. So this gentleman in our lab, Hong Shen, went ahead and he cloned every single gene of the herpes genome into a GBM cell line. And then he took, and that was about 70 or 80 genes, and then he took NK cells and he did a killing assay. And he said, which of these genes are the NK cells getting activated and killing the GBM, and which ones are they being inhibited by the GBM? And it turned out there were about 3 of the latter, but about 5 of the former. They found about 5 genes that you put that single gene in a GBM, all this messy stuff, and you tilt the scale, and that GBM goes to history with the NK cell. So he found the genes in the herpes virus, and I'm going to refer to 2 called glycoprotein E and glycoprotein I. And these 2 were the most potent. If you have GE-GI, GI by itself did nothing, but GE with GI or GE alone, you get the killing. And the killing would release the granzymes, you get the cell death. So I'm just going to explain to you how this works and how we're exploiting it. So like I said, normally what happens is in the top you have your NK cell, and there's its Cd16 sticking off at the surface. It binds FC. And of course, your tumor cell has tumor-specific immune globulin, Rituxan, Herceptin, that sees either CD20 or HER2, and it, on the antigen-specific way, the FAB fragment binds to that specific antigen. And the NK cell, then binds to a different region. It binds to the FC region here. As you can see, the FC, the non-antigen region. And it locks and loads, and when you get dimerization, shoots a signal to the NK cell, it kills the tumor cell. And that's at least in part how things like Herceptin and Rituxan are working in an anti-tumor. It's called antibody-dependent cellular cytotoxicity. It's been known for 40 years. What we found was something completely different and relevant to herpes and other pathogens that I'm not going to talk about. But so there's your CD16, and it's binding to that FC portion of the antibody here. But it turns out the GE and GI of the herpes genome when it infects a GBM or whatever cell it's infecting, when that GE and GI go on the surface, that forms a protein that also binds FC. It's called an FC binding protein. So just like Cd16, it's binding FC, but it's binding it in a different region. And it allows the Cd16 to still bind when immunoglobulin is bound to the FC binding protein, or the dimer up here of Cd16 with any immunoglobulin on its surface. There's the immunoglobulin in your body. It turns out, I'll show you, NK cells are covered with your immunoglobulin. And without any antigen specificity, your innate immune system working before you have any antigen specificity, it binds here. It finds this receptor on the surface of the HSV-infected GBM. And then they lock and load. And again, the key with this is this without any antigen specificity. So before you have immunity to herpes with B cells and T cells, your NK cells, which are floating around with immunoglobulins stuck to their CD16, use that dimer to see FC binding proteins. And I'm not going to talk about it today, but FC binding proteins are also on Staph A and they're on strep. So it's a broad mechanism of innate immune recognition of pathogens. Any pathogen that encodes an FC binding protein is recognized by this mechanism. And then you get your lysis. And this crystallographic structure of this region was known, CD16 binding to human FC, and of this, FC binding proteins bind to human FC. And as it's shown here in the in silico representations, here's GEGI from herpes, here's CD16 from the NK cell. No overlap whatsoever. So this can and does occur structurally, and we have lots of proof of this. I'm just going to show you a couple things. Here's how this would work in our cartoon. So now you got your GBM here in the midst of your normal tissue. And again, your HSV is coming in. And because of that tumor-specific promoter, it can bind only to, it infects and will become lytic in only those cells. And so the first cell, goes to lyse, but it can't because the NK cell comes in quickly and lyses that HSV-infected GBM before it can really spread throughout the whole tumor. So that's lyse. It goes to lyse the next one. You notice there's no spreading of the virus. Here's it up close of what's going. There's the GBM membrane, HSV infected, so there's GE. HSV infected, there's GI. They come together and form a high affinity FC binding protein for immunoglobulin. Then the NK cell, which is floating around up there with its, you'll see it's popping up all over the surface of the GBM here. The NK cell has CD16 on its surface. It's got immunoglobulin stuck to it. And it has, as I said, this binding area called the bridge area binds the CD16, and this is called CH2-CH3 region that binds the high affinity FC binding protein. And then you get your dimerization, your activation, and your killing. So, is this real? So here we're looking now, we're staining NK cells to say, do they have immunoglobulin on the surface when they're floating around in your blood? And the answer is they do. Here's immunoglobulin staining, here's your NK cell, here's your T cell. You can see the drastic difference. And that's what it would look like. But it turns out we're all different. We all have different affinity. So here's 3 different donors in addition to this donor. You see, look at the numbers here. This is just indicating the amount of immunoglobulin that's binding. Relatively high, low, low. So depending on who you are is how much immunoglobulin is on the surface of your NK cells. If that's the case, then, and if the killing mechanism I showed you is the case, then there should be variation between all of us when we actually see a herpes-infected cell, whether it's GBM or otherwise. There should be a difference because we have only so much immunoglobulin on the surface, or abundant immunoglobulin on the surface of the NK through the CD16. And so what we did is we took 25 people randomly and just measured their intensity. How much immunoglobulin do they have on their surface? And then just asked ourselves, do they all kill GBM the same, or is it different? Strikingly different, highly correlative. So the more immunoglobulin you have on your surface, the more activation or killing you have of the GBM. Perfect correlation. You take that same 25 people and you have them kill an NK-sensitive target, K562, that doesn't have GEGI on its surface. No correlation whatsoever, same people. So that told us that, in fact, this is a functional mechanism that's relevant to people. And you can understand, maybe begin to understand why, at least in this case, why different people respond to different pathogens differently in this case. This, by the way, is called, we've got a name for this. It's called FC bridging ADCC. So because the FC fragment is bridging between the NK cell and the herpes infected cell. So how about in vivo? Does FC bridging cellular cytotoxicity protect it? Because we know mice have Cd16, they have NK cells, they bind their own immunoglobulin. This should be able to see, we infect the mouse with herpes, the NK cells should see it and prevent it. Well, it turns out that this is a human pathogen. And this FC binding protein doesn't see the mouse CH2, CH3 region. So in fact, you don't get recognition and you get 100% fatality. But we should be able to take that same mouse and inject it with human immunoglobulin. And if there's enough, it will compete for the CD16. It does bind human immunoglobulin. Mouse CD16 binds human immunoglobulin. And we know that CH2, CH3, this whole complex recognizes the FC binding protein. So now if I give one shot of this antibody, and it could be an irrelevant antibody, it should provide protection when I give a fatal dose of the HSV-1. And in fact, I should be able to cleave off the FAB fragment and just give FC, because this is a complete innate immune function. And indeed, that's in fact what is the case. As you can see here, whether I give Rituxan, whether I give daratumumab, or whether I just give the FC portion of any immunoglobulin, you get nearly complete or complete protection against the HSV infection, fatal infection in the mice. Further, if you knock out the NK cells, as shown in the bottom line with something called acylo-GM1, before you do the exact same experiment, you completely lose the protection. Because now, even though you're given the FC fragment before you give the herpes, you've taken out the NK cell, that CD16 is not there. And so even though the FC fragment is binding to the herpes FC binding protein, there's no killer cell to come in and clear it out. So therapeutically, what could we do to delay those NK cells from killing that GBM? And it turns out that there's a natural bacteria pathogen, that Strep pyogenes, that makes an endopeptidase that cleaves your immunoglobulin. And this actually has been given in vivo to patients, this endopeptidase, to stop hyperacute rejection and kidney transplants. And in vivo, in the human body, it cleaves this. So you're just left with this piece, which binds to CD16, or this piece, which binds to that FC binding protein. And so if you purify this, you should be able now to take your cartoon, and what you should do is be able to inject this FC piece, and it should bind only to the herpes-infected FC binding protein and block the NK cell from seeing that free site. Here's the FC fragment coming in, blocking GEGI. So now, when the NK cell comes by, that site's occupied. It can't get at that cell. The virus has time to spread, replicate, and lyse, and you can prolong your survival. And so we've been working on this to do this in vivo and ultimately in patients for this and other pathogenic diseases that I'm not going to talk about today that are relevant to this mechanism of immune surveillance, when we made another discovery. So one thing is our lab is developing EGFR CAR for GBM. I'm not going to talk about it, but it's relevant to what I'm going to tell you about. And it turns out you can get a nice little bump in survival in xenogeneic human GBM when you take NK cells and express a CAR on the surface that can find both mutant and wild-type EGFR, and this CAR that we have can. It turns out that if you give HSV and NK CAR sequentially, you can get this, actually start to get a survival curve with these mice. But what we found is that we took our original herpes virus, which we now have in patients to start with our clinical trial, and we actually added to that herpes virus an inhibitory ligand that finds the NK cell cure-like molecules that I was telling you about. It's called KLRG1. So we added this E-cadherin to the herpes virus. When it infects the GBM, it secretes a ligand, it expresses a ligand that keeps the NK cell from killing it. So it actually, without needing to do that difficult cleaving experiment I talked about, if you change your herpetic virus to express an inhibitory ligand of NK cell, it holds them off for a bit. And we've now shown that actually just by itself, this virus can be curative in these models. So we're now scaling up to move this virus to the clinic. In the meantime, this is just the cell. In the meantime, we started our first clinical trial for a PO1 we have with our original herpes virus that is specific for GBM. And the idea is that we will next do GBM with EGFR CAR, and then ultimately we will combine the HSV1 for GBM followed by a NK CAR therapy, or if we have it in time, we will move to our treating recurrent GBM with our oncolytic herpes virus that expresses E-cadherin and then combine with the EGFR CAR NK cells. So I'm going to stop there. I wanted to talk about today are cytokines and NK development, specifically the role of IL-15, how we use receptor biology to find where the precursor is, where it traffics to, where NK cells develop. We talked a little bit about some of the malignant counterparts to those. We talked about the human NK cell subsets and their role in the immunoregulation of the immune system prior to antigen-specific immune responses. And then clinical application of receptor biology, understanding how NK cells see herpes virus, trying to prevent that to prolong oncolytic herpes virus from working in GBM, and then ultimately combining that with an NK cell CAR in this particularly terrible disease. So I'll stop there. Thank all the people in my lab, 15 of whom moved with us to City of Hope. This is the real mastermind here, Jinhua Yu, and Hongshen Dai is back there. Jinhua is really responsible for a lot of the work in our laboratories. So thank you very much for your time. It's such an honor to be here. I appreciate it.